Hi, y'all. I'm Calvin here to talk about how the kidney works. Complicated organ indeed, but any complex concept can be put in simple terms. To understand the kidney, we have to learn something called osmosis. Osmosis is the idea that electrolytes like sodium and potassium, being electrically charged, can form electrostatic attractive chemical bonds with water molecules, which means they can attach to water molecules and pull the water molecules wherever they go, even across semi-permeable membranes. So the working mechanism of the kidney can be explained with a beaker divided by into two by a semi-permeable membrane. So what we do is we fill the beaker with water and then divide it into two with a semi-permeable membrane. And since due to gravity, both the sides of the beaker will be equal with water. The first step in the process is on one side of the beaker we throw some sodium ions. For example, like salt, we throw salt and dilute it, and the salt gets divided into sodium and chloride. And sodium, what it does is it attracts the water molecules from the other side of the beaker through the semi-permeable membrane. And on the left side, the water level increases compared to the right side. We add pressure with a piston pump from the left side. We use a pump to add pressure manually, physically, and the pressure causes the water molecules and the soda ions to move towards the right side through the semi-permeable membrane. It pushes the ions according to Pascal's law, not Pascal's law. According to osmotic law, not even osmotic law, this is reverse osmosis. Reverse osmosis, what we do is we give pressure on the other side to increase the flow of water molecules and ions, sodium ions from left to right. Now we do some microscopic engineering, and what we do is to this apparatus we add a specific pump on the semi-permeable membrane, which only sends the sodium ions to the opposite side. So what does the sodium ion pump do? It is a sodium ions importer. From the left side, all the sodium ions one by one they go to the right side. These electrolytes, such as sodium, potassium, they have the power to pull the water molecule to the side which they swim across. So the sodium ions also pull the H2O molecules to the right side, increasing the water level further on the right side. The exact working mechanism of the kidney is given by this experiment. The semi-permeable membrane denotes the gap between the kidney lumen and the blood vessels which carry the blood and the kidney fluid. It is also separated by the transcellular fluid in the interstitial space, which is of an advanced level. So the semi-permeable membrane separates the blood and the kidney lumen, which is the nephron lumen. The sodium ions is from the water we consume and the food we eat and the salt we consume. The pumps, this micro pumps on the semi-permeable membrane are transcellular pumps which are present on the cell walls of the nephrons and the kidney filtering unit, which is the nephron. And these are of various types like sodium transporters, potassium transporters, co-transporters, 2-ion transporters, importers. They carry one or two ions from the kidney lumen or nephron lumen to the arterioles and arterioles to the lumen. So this flux of ions from one side to the other side is guided by this pump, and these pumps carry ions to the other side, and also these ions carry the water molecules to the other side, increasing the water level on the other side. So the piston pump in the diagram denotes the blood pressure from the heart. It forces a blood pressure on the surface of the surface of the nephron, and the filtration happens due to this pump mechanism also. So three mechanisms are involved. The first mechanism is the osmotic pressure from one side to the other. Second, it is the piston pump pressure, which is the blood pressure. And third, the ion transporter pump, which is the sodium pump, 2-ion transporters, 3-ion transporters, co-transporters, and symporters, depending on the ions they transport from one side to the other. 30% of the cardiac output goes to the kidney. Thus, in a day, 30% of the heart's blood output goes to the kidney. That means 180 liters of blood is filtered in a day. The human body has only 5 liters of blood. Thus, most of this 180 liters of blood is reabsorbed by the arterioles in the kidney. To explain the function of the kidney 3 does three main things. One, it senses and maintains blood pressure. Second, it excretes the waste from the blood. Third, it produces erythropoietin, which is required for hemoglobin synthesis, which runs the blood. The blood pressure sensing apparatus is called the macular apparatus. I'll show it to you very soon. The excretion of waste products is done by the 1.2 billion nephrons and the urinary bladder. The nephrons filter the blood and the urinary bladder collects the urine. And the connection between the nephron and the urinary bladder is given by the ureter. Erythropoietin production used for heme production, hemoglobin production, is for another day. It is a complicated subject involving a lot of biochemistry. Sketch of the kidney, urinary bladder, and the urethra. The tubes which connect the kidney and the urinary bladder is the ureter, and the urethra excretes the urine from the urinary bladder when wanted or sometimes even unwanted in some pathological conditions. So now I will be dealing alone with how the kidney works. So there are 1.2 million nephrons in the kidney. These nephrons, they collect the blood from the arteries, renal arteries, which are branches of directly from the aorta. These renal arteries, they sit directly in the glomerular basement membrane cup, and the nephron excretes the waste, re-gives the fluid to the blood, and extreme wastes are alone given out through the ureter. Renal artery from the aorta gives the blood to the kidney, nephron filters it, and renal veins takes back the blood. Waste from the kidney is given out through the ureter to the urinary bladder. Filtering apparatus of the kidney has calyces and pyramids. The calyces contain the collecting ducts, and the pyramids, they contain the nephrons. There are multiple nephrons, like millions of nephrons in each kidney. The blood from the aorta is given out to the renal artery, and the renal artery branches out and supplies the entire kidney, and the arterioles from the branches of the arteries, these arterioles, they supply the nephrons, each and every one of the 1.2 million nephrons in each kidney. So the arterioles are so small that they can supply the Bowman's capsule of each and every nephron, which is 1.2 million in number. There are two types of nephrons, cortical nephrons and juxtamedullary nephrons. So depending upon the depth of the nephrons, the more ions they will collect, the more waste they will collect. Juxtamedullary nephrons, they collect more waste products from the kidney, from the blood. Now I have exaggerated the size of one nephron to show its structure, which is orange in color in this picture. Here is a more clearer picture, and the green part in this picture is the macular apparatus. This is the macula densa. It senses the blood pressure from the arteriole, which calculates the entire body's blood pressure, and it sends information to the lungs, where angiotensin II receptors are present, and the lungs determine whether there is a lot of fluid in the lungs or not. And depending upon the lung fluid volume, the blood pressure of the heart is regulated by regulating the blood volume. And how is the blood volume regulated? By excreting more water, the blood pressure will come down. So that is how the macula densa apparatus regulates the blood pressure. Now we have isolated the nephron, and looking at its structure, it has the following parts: Bowman's capsule, loop of Henle, and collecting duct. The Bowman capsule filters the blood from the arterioles. Loop of Henle, it has the ion pumps. The ion pumps, which transfer the ions from outside to the inside of the lumen of the nephron, and the collecting duct collects the urine. And in this picture in green, we have included the macula densa apparatus, which regulates the blood pressure, or at least senses the blood pressure. Blood from the heart through the aorta to the renal artery and into the arterioles and into the basement membrane of the Bowman's capsule. This is how the structure looks. An afferent arteriole up and efferent arteriole below, They go through a circuit of arteries and they enter and get filtered. The blood gets filtered and the fluid alone goes through the Bowman's capsule. Not only the Bowman's capsule, the entire nephron is surrounded by arterioles. The orange part of this diagram is the nephron and the red part is the arterioles which surround it. Proximal convoluted tubule after the Bowman's capsule, which can be called the PCT, it is the part which excretes most of the sodium ions. Thus, the sodium ions pulls the water molecules through the nephron to the blood vessel, which is shown in red. Most 90% of the blood which is filtered goes back to the blood arteriole through the proximal convoluted tubule itself. Loop of Henle goes into the structural depths of the kidney. It adds the length to the nephron. The nephrons are long and are folded, and most of the folded part is the loop of Henle, which is one fold, and the walls of the loop of Henle has a lot of cells which contains pumps. These pumps transfer sodium, potassium, other ions and water, bicarbonate from one side to the other side, which is from the lumen of the nephron to the blood vessel, which is the arteriole, shown in red and orange. The space between the arteriole and the loop of Henle, as we go deeper and deeper into the kidney and lower through the loop of Henle, this space contains more ions, more waste, and the blood from the arterioles which go deep into the kidney, they collect more waste because they have a slow speed. The slow flowing blood collects more ions and their tonicity is higher. The normal tonicity of the body, that is the normal ion concentration of the body, is 300 osmoles, exactly 290 osmoles. And here the tonicity, it increases from 300 to 1200 osmoles as we go deeper and deeper into the kidney and lower through the loop of Henle. Again, just like the pumps, sodium pumps and chloride pumps and other pumps through the semipermeable membrane, as you go deeper and deeper into the kidney, there is more ions to transfer from one side to the other side. Thus, more ions and more waste from the arterial blood gets transferred to the nephrons as you go lower and lower through the nephron loop of Henle. As we move upward from the lowest point of the loop of Henle, from 1200 osmoles back to 290 osmoles, all the osmotic waste products should go back to the blood due to the osmolarity, right? But there is an impermeable part which is called thick ascending limb, which does not allow the waste products to go back to the blood. Thus, all the waste products remain in the lumen of the nephron, and thus it goes to the collecting duct. The waste goes to the collecting duct into the ureter. Thus, the concept of piston is given by blood pressure in the Bowman's capsule, and then the concept of osmosis takes place in the proximal convoluted tubule, and reverse osmosis through the pumps in the cell wall happens in the descending limb of the loop of Henle, and impermeability and retention of urine or waste products, waste ions, happen in the thick ascending limb of the loop of Henle, and distal convoluted tubules have specific ion channels where reverse osmosis happens again, where a lot of hydrogen ions are released back into the nephron from the blood, and the collecting duct contains the ultimate urine. From the collecting duct thus flow into the pelvis of the kidney, and from the pelvis of the kidney it goes to the ureter, and from the ureter it goes to the urinary bladder, where at the end of the urinary bladder there is a sphincter which is voluntary, which controls the movement of urine outside the body. Thank you so much for finishing the video. Thank you so much for finishing the video.